Thank you, Sarah, and I'd like to thank the meeting organizers for the opportunity to give you an update on the latest developments at RhinoCyte. We are a private biotechnology company, and we are seeking to change the way that neurodegenerative diseases are treated. We are advancing treatment paradigms to leverage our cell therapies for high unmet clinical needs, including spinal cord, our first product, and Parkinson's disease. We believe that the RhinoCyte technology affords patients a novel therapy that extends and enhances the quality of human life without immunosuppression or genetic manipulation. We are looking at harvesting the cells from the rhinoepithelium, hence the name RhinoCyte. The inventor and the chief scientific officer, Dr. Fred Roizen, discovered the tissue years ago and was intrigued by the high lifelong regenerative capacity of the tissue, the fact that it contained stem cells, that it was readily accessible from a biopsy perspective, and that it is autologous in terms of not only biopsy but reimplantation. He discovered that the types of cells that can be replaced with the cells that he is culturing are motor and sensory neurons, oligodendrocytes, and other support cells. We are looking at spinal cord injury, as I mentioned, for our first product. Quickly, as a follow-on, is Parkinson's disease, and then we have a number of other opportunities within the technology platform. So what makes the RhinoCyte approach unique? We have an excellent safety profile and an ease of harvesting the cell. We have over 150 patient lines that we've monitored for over 3 years in culture. The cells respond to unique signals within the environment and do not require genetic engineering. The RhinoCytes, we believe, synthesize molecules that promote neural cell proliferation, survival, and differentiation. And we have seen efficacy demonstrated in several animal models in spinal cord injury, as well as a Parkinsonian model as well. Key attributes for the RhinoCytes include the autologous sourcing, the fact that we will produce them in a cGMP manufacturing facility. One of the big advantages, if you're familiar with the hospital environment, is that there's no downstream processing once it gets to the administering office. They are cryopreserved, and that gives us flexibility in terms of scheduling and administration. And as I mentioned, there is no immunosuppression, which is particularly important for the spinal cord injured patient. Unique properties of these RhinoCytes include that they're nestin and perforin positive, as well as beta-tubulin 3 positive. We believe that they are neuroprogenitors with key markers of neural lineage. They do express telomerase, and there's low apoptotic activity regardless of the number of passages. In terms of pharmacological activity of the RhinoCytes, they do, in the spinal cord injury lesion, lose the reactivity to nestin and mature to the neuronal bells that do express the beta-tubulin 3, as I mentioned, and they do not become astrocytes or contribute to glial scarring in this model. In terms of myelination, an important factor in spinal cord injury, the RhinoCytes do induce a higher degree of small axon remyelination, and that's going to be something that we're going to look at closely as we look at the spinal cord injury trial. This is our first product, as I mentioned. We have validated animal models in both the contusion and the hemisection. The market opportunity is about 1.3 million patients with 13,000 new cases annually in the U.S. alone. The cost-effective treatment is of particular interest when we look at reimbursement because there is over $40 billion in annual treatment cost for these patients. So this type of treatment will be very cost-effective when compared to the treatment cost if we're able to make a slight improvement that will be meaningful to quality of life for these patients. We have been awarded an orphan drug designation for spinal cord injury, so that does give us expedited review and some other opportunities for funding as well. In terms of our IND process and timing, we have completed the pivotal toxicology and biodistribution studies. We are in final qualification of a cGMP facility that is next to the university and the founders of the company. We have completed the majority of the analysis and IND submission paperwork. We are now working on the CMC section and anticipate in the third quarter of 2014 having that into the FDA, and that will lead to the phase 1 preparation in the following quarter. Our regulatory team is headed by Dr. Joy Cavaniero. She's our chief strategist and was one of the architects in putting together the CBER division when she was at the FDA. We also have Dr. Costello and Dr. Williams in helping us put together the CMC and the document for submission, respectively. Our phase 1/2 multi-center trial for spinal cord injury is open label. A total of 20 patients and 3 centers are planned. We're looking at subacute, functionally complete T3 to T12 lesions in the spinal cord injured patients. We're looking at transplantation 120 days post-injury, and the reason for that is to allow spontaneous recovery to complete before we give the treatment. Primary endpoints, of course, are safety, but we do have secondary endpoints, multiple opportunities for outcome measurements for efficacy. We are going to be following the patients long-term with frequent short and long-term MRIs. And our clinical team in helping us prepare the final protocol is headed by Dr. Jonathan Hodes, our Chief Medical Officer. He is an experienced neurosurgeon, and he will be working with the other centers in finalizing their input on the protocol. Dr. Jane Lepkowski has been working with us on putting the protocol and the clinical plan together, as well as Madeline Marino on the quality assessment as we put the product into the cGMP facility. After Parkinson's disease, we're quickly followed by Parkinson's. We have an assessment that this is one of the disease states that might be ideal for this type of product. One reason for that is the loss of one specific type of neuron, the dopaminergic neuron, and we would replace that directly with the cells. There's no lasting effective therapy right now on the market. There are pharmaceutical interventions, but they are not a cure for the disease, and over time they lose their effectiveness with the patient. RhinoCytes, we have found, can form the dopaminergic neurons that are needed in the patient type, and there are 1.5 million patients currently in the U.S. with 60,000 new cases each year. The real cost analysis benefit is that over $25 billion is spent each year in care for these patients. So again, when we look at this model, an opportunity for this type of product to either cure or mitigate the disease would be very much in favor from a reimbursement perspective. The one thing that we did find in the studies that we did, surprisingly, is these cells do not need genetic engineering. Steps for PD for the clinic include the preclinical meeting with the FDA, which would be upcoming next year. We have several external collaborators that have expressed interest in working with us and helping us with the technology. Our pivotal studies are scheduled for the third quarter of 2014 with an IND submission the following year. We do have a robust intellectual property portfolio. We have both cells and use patents in the U.S. and in Europe, as well as Israel and Australia. We also have filed for PD and diabetes as single patents in addition to the overriding umbrella patent. Our leadership includes our founder and chief scientific officer, Dr. Fred Roizen, myself and Steve Gaylor, who is a founder of the company as well, and our chief advisor. We do expect growth potential and opportunities from the platform. I've spoken about spinal cord injury and Parkinson's. We do look for other opportunities as well in potentially type 1 diabetes as well as ALS. In terms of our financial status, we have raised $5 million to date in a Series A, as well as $4 million in public funds from the NIH to support the development of the technology. We are currently looking at $10 million targeted for Series B. The first tranche is closing very shortly, and the investors from this first tranche include the University of Louisville Foundation, Commonwealth Seed Capital, Kentucky Seed Capital, and a number of regional angels that have actually invested in the company from its founding. Use of funds include the IND submission, planning of the phase 1 trial for spinal cord injury, and final qualification of the manufacturing facility. We will have sample product for the IND submission from that qualified facility, as well as production of the phase 1 material. In terms of the Series B, the second tranche is $8.5 million, and we will begin initiating fundraising for that third quarter of next year. The use of those funds would be completion of the spinal cord injury phase 1/2 trial and further development of the technical platform. I'll be available afterwards for any questions that you may have or any comments or helpful suggestions. Thank you.